I think now we can get started. Hopefully we can finish this chapter for the wastewater treatment this class. Okay, so we can jump into some more interesting contents in air pollution. This is the part 3 of this wastewater treatment system. And this class, we're going to focus on the aeration tank or the activated sludge system. Okay, so I'll just use these names interchangeably because tank. So basically, what happens in a aeration tank is we have a lot of bubbles, right? Generate a lot of bubbles and then let the bacteria to propagate themselves, right? So that we can remove the BOD or the organics from the wastewater. And the aeration tank is categorized as the secondary treatment process. Okay. So the reason why we call it activated sludge is because in order to make these aeration tank to work, we need to provide some bacteria inside. Basically, those are typically the same bacteria that's existing in rivers or in lakes, which can decompose the organics, right? So if we just draw out the entire process for this secondary treatment or the aeration tank process, what happens is we introduce the pre-processed wastewater that's coming out of a primary treatment or primary clarifier. And then we're going to mix some bacteria with the wastewater. And then after the aeration tank, basically to grow these bacteria to consume the organics or the BOD, we introduce the effluent into a secondary clarifier. Okay, so basically we further remove the solids. So mainly these solids are just grown bacteria because they enjoyed a lot of food in here, and we also provided unlimited oxygen, right? So they can reduce the concentration of the organics. So basically, most of the solids are just the bacteria itself, right? So what that means is while we remove the organics, the water is going to get cleaner. And then basically this is the effluent, which is further treated from the secondary treatment process. But with the sedimented solids or the solids that settle down to the base, we have to use them or we have to remove them. Otherwise, the solids are going to grow higher and higher, right? We have to process it. So for these solids, as we said, a lot of them are just bacteria, right? So instead of directly dump it into the landfill, we created a line that's basically returning this sludge or these solids back into the inlet of the aeration tank. So basically we call this as the return sludge. Okay, of course, we're not going to use all of the bacteria. So some of them will just go to the waste. Right. So if you analyze what is the flow that's involved in this system, right? So if you draw let's say a control volume here. Okay. So if you analyze what's the inflow, the inflow is just going to be the flow coming out of the aeration tank, right? So if I want to write out this flow rate, okay. So this inflow is further going to be equal to the wastewater flow that's at the inlet from the primary treatment, and also the return sludge flow, right? So these two adding up together, that's going to be the inflow here. And then further, we have the effluent, and also a fraction is the waste. Okay, so maybe here I should use a, let's say Q0 to indicate what is the flow that's coming out of the primary clarifier. Okay, so this is trying to analyze the mass balance. So we're going to use this concept further when we design the activated sludge or active aeration tank system later on this class. So now you can see why we also call this process as the activated sludge process. It's mainly because these sludge here, they're activated. They're not dead materials. They're not just rock or sand. They are bacteria. Okay, so that's why we also call this process as the activated sludge process. All right. So this is just a brief introduction of what we're going to learn. But again, if we do a quick recap of our last class. So last class, we mainly spent some time on introducing the on-site treatment process, right? So on-site, we have the very simple type. It's a septic tank, right? It has 2 compartments. And then we spend more time to introduce each component of the wastewater treatment plant in more details. For example, we have the bar racks or the bar screen. We have the grit chamber to remove the particles with medium size using the spinning movement of the wastewater flow. You have the comminutors, which is cutting these larger chunks of solids into smaller sizes. So people also use the equalization tank, which is to moderate or to buffer the different or the large variation of wastewater flow, so that you don't the water treatment plant doesn't need to deal with a large fluctuation of the flow. So in terms of the primary treatment, we said that the major component is basically the primary clarifier, which is using the sedimentation concept to remove the particles. So again, regarding the sedimentation, we introduced that in the water treatment or the drinking water treatment, that when it's using this gravity, we can define a parameter called the overflow rate, right? So overflow rate is basically the sedimentation flow rate. And that can be calculated by the flow rate going into this tank or this reactor divided by its surface area. Okay, so this is how we calculate the overflow rate, or how we can use this overflow rate to calculate the other parameters. And at the same time, we define a characteristic time, residence time, retention time, detention time, right? So that is calculated by volume divided by Q. Okay, so this is basically the 2 equations that determine the parameter or the performance of these primary clarifiers. So in terms of the secondary treatment, as we said, it's mainly involving the aeration tank. And of course, after the aeration tank, we're going to use the secondary clarifier. Right, the secondary clarifier is used to remove the suspended solids, which is mainly the bacteria. And we also call this process as the activated sludge process. Okay, so now if we spend more time on this activated sludge process, so last class, I think we also introduced different categories of the metabolism. So it's basically using the metabolism of the bacteria or the microorganisms to decompose these high BOD values, right? To remove the organics. So the metabolism can be divided into catabolism and anabolism. So the catabolism is mainly just converting these organics into energy. And a lot of time it's just heat, right? To sustain itself. Basically, as a human being, we eat food, right? So to support our energy consumption, for example, if we run, if we walk, if we sit and study, right? So those are going to consume energy. But at the same time, when we ingest these food, we're also converting a fraction of food into our body cells. Okay, so that process is called the anabolism. So basically, it supports the cell growth. Right, so it's synthesizing process. So because of that, the let's say the aeration tank or the activated sludge process is going to use these 2 processes, and people have to design or to find the bacteria that can do balance between these 2, right? Because on the one hand, the catabolism is quite important because it determines how much food the bacteria is consuming. But at the same time, we want these bacteria to propagate themselves, to grow more cells, so that we don't need to cost a lot in terms of transporting these bacteria, right? So let's imagine if we just put in hundreds of bacteria. So if they can propagate into millions in a time period of hours, right? So that's going to boost up the BOD consumption much faster. But at the same time, we want these bacteria to consume food, right? Just based on itself, not basically using all of the energy to propagate, but also to consume these organics. Okay. So further, in terms of these consumption process, I think we introduced them briefly in the ecosystem, right? So there are different consumption mechanisms. So there can be aerobic decomposition. So aerobic decomposition basically involves oxygen. So it's like animal cells, right? So when the animals decompose these organics, they need oxygen. And the major reason for this aerobic decomposition to be dominant process is that it can produce a large amount of energy, right? So, and it is actually most of the time it's a natural water self-purification process. So what happens in rivers or lakes, it's also happening in this activated sludge process. So it needs molecular oxygen. And in terms of the end products, it produces carbon dioxide and water. And at the same time, it releases a large amount of energy. And because of this large amount of energy, that can lead to a high growth rate. Okay, so the bacteria can do more anabolism from the aerobic decomposition. And basically that means it's going to produce more sludge. Okay, just recall here, we call this solids here as sludge, right? So if they can propagate more, then it means that we're going to have more sludge. So it has a high reaction rate and less odor, right? So that's why we like this process. And actually in wastewater treatment, it is a dominant process to remove the BOD and also to grow these microorganism cells. So it's not very suitable for very high strength wastewater, which has a very, very high BOD. Okay, if it's, let's say, if the BOD is higher than 10,000, right, milligram per liter, so that's going to consume a lot of oxygen, and it's difficult to maintain this oxygen concentration because we know that the maximum dissolved oxygen under normal temperature is just around 8 to 8.5 milligram per liter. Okay, so what this means is that we have to use a very strong bubbling system to introduce the oxygen, which is quite difficult to manage. So if it has a very high concentration here, then maybe instead of directly introducing these wastewater into the aeration tank, there can be dilution or there can be some other pre-treatment process to lower down the BOD values. So this is the aerobic decomposition, which is the most important decomposition method. So there are also two more. So one another is the anoxic decomposition. So if you look at this NO here, so if it involves anoxic, it generally refers to the nitrogenous species. So which is reducing the nitrogen, because we know that nitrogen is a nutrient, right? So if the wastewater contains a lot of nitrogen, and then if we don't deal with it, then it's going to cause the algae bloom and cause some other environmental hazards. So for the wastewater that has a very high concentration of nitrogen, we have to go through this anoxic decomposition. So basically, the microorganisms are going to use nitrate or nitrite as the electron acceptor. So instead of using oxygen to support this metabolism process, they're going to use either nitrate or nitrite. Sometimes they can use ammonia as well, right? So basically, what comes out of it are going to be nitrogen and carbon dioxide and water. So you see that this is also pretty clean. So for this process, it also releases much energy, a lot of energy, and also it produces relatively high biomass production. So we're going to have a high sludge concentration as well. But as you can see, anoxic decomposition is quite different from the aerobic decomposition, right? And if it's reflected in the wastewater treatment, then basically people have to use different types of bacteria, right? So for these bacteria, these are just natural water bacteria. But for this anoxic decomposition, we have to use some other bacteria, which is less common in the environment, right? So because of that, this process is also going to be a little bit more expensive because we're dealing with other types of bacteria. So it's used to remove nitrate, nitrite, and this is also called the denitrification process. Okay, it's to lower down the concentration of the nitrogen in the wastewater. So the last one is the anaerobic decomposition, or people call it fermentation, right? So this is similar to the process where we generate, we produce yogurt from milk, right? So basically we add in some bacteria to let it do this anaerobic decomposition. So this process is also quite slow. Just imagine if you want to make yogurt, it's going to take a few days. But if you directly expose the milk to the normal environment, let's say a glass of milk to the air, maybe after a day, the milk is going to go bad because some bacteria can directly use oxygen, right? That's going to be a much faster process. And then they can propagate very quickly. Milk, let's say the surface can turn green, right? It's because of, you can even see some colonies on that. So the anaerobic decomposition or the fermentation process is normally quite slow. I have to label it here, okay? And also it produces less energy. So then you may wonder, why do people still use it, right? Why do we still use the fermentation process? So the reason why we use it is it can generate a very important byproduct, which is a methane gas, okay? So let's go from the beginning. So for the anaerobic decomposition, so it has to happen when oxygen or nitrate or nitrite are not present, okay? So if there is oxygen, then the bacteria are definitely going to go through the aerobic decomposition because that's a preferred way. It's a fast process. It's also a process that generates a lot of energy. So for this anaerobic decomposition, the bacteria normally use sulfate ions, carbon dioxide, and organic matter. For example, some organics like glucose as the electron acceptors. So basically, they're going to consume these species. So for this anaerobic decomposition, it's low biological energy production. It has very slow growth. But the reason why we use it is because it can generate methane gas. We can use that for combustion, right? We can use that for heating or cooking. So for some remote, let's say rural areas, people will use these fermentation tanks to generate methane gas and then further burn it as a fuel. But for this fermentation process, anaerobic decomposition process, it's very sensitive to temperature and the loading change. So basically, the loading here means the BOD loading, right? So if you have different types of organics, then the fermentation will be quite different. So it requires a high maintenance because of these requirements, and it's not suitable for very low strength wastewater. So where the water has a low BOD. So if it has a low BOD, then because it's a slow process and also because it produces less energy, the bacteria are not going to propagate themselves. So the processing is not going to be very efficient. So the detailed steps for this anaerobic decomposition is that first the organic matter is converted into acetic acids, and then the acetic acids are further converted into the methane gas. So I have to say that the methane gas or these fuels coming out of fermentation is a major reason why we use the anaerobic decomposition. So because of that, in the industrial or the municipal wastewater treatment plant, we never use this anaerobic decomposition, right? So now if I give you a quiz question, why is anaerobic bacteria grow faster than the anaerobic bacteria? So we'll get 10 more seconds. Okay, we'll stop here. All right, so most of you guys are correct, right? So the reason why we use aerobic bacteria is that the aerobic decomposition produce much more energy, okay? All right. Okay, so now we can try to talk about the design of this aeration tank or the activated sludge process. Okay, so if you look at where this process is taking place, again, this is a schematic diagram of the wastewater treatment plant, right? So the secondary treatment or the aeration tank or the activated sludge process is here. Okay, so it's after the primary treatment where you have the primary clarifier, right? So basically, it's going to be the pre-treated wastewater. The reason why we don't move this section here is because, as we said, the sewage in has a lot of large chunk of solid, right? So we don't want these solid to cover or to interfere with the bacteria's growth. So we first need to remove these larger solids and then send these relatively cleaner wastewater into this, maybe we can also call it bioreactor, right? We can basically the biological species or biological reactions are taking place in this aeration tank. So there are several definitions for this process. So first, the aeration tank, as we said, it's basically just this reactor here. Okay, so this is a reactor. It's composed of bacteria and pollutants. So here specifically, these are the BOD generating species or the organics, right? So what happens inside is the mixing of the water and the aeration process. We bubble a lot of air from the bottom, right? We introduce a lot of oxygen here. So that's why we can also imagine it as the bacteria are enjoying their unlimited food, right? We're introducing a high concentration of organics. We also give it unlimited amount of oxygen. So it's always have enough oxygen there. So in terms of the second stage of this secondary treatment, we have the, excuse me. So we have the secondary clarifier, which is downstream of this aeration tank. Okay, so the reason why we need to set up this secondary clarifier or secondary settling tank is that while the bacteria growing here, they're going to form aggregates. Let's say this is a single bacteria floating in the organics, right? So while they populate, when they get more of them, they're going to aggregate. They're going to collide with each other and form larger chunks of bacteria, or you can also call them as colonies, right? So when their mass grow larger, they're going to basically settle down onto the ground, right? So we can use this way to remove these larger bacteria or these suspended solids down to the base of the clarifier. Okay, so this is the secondary clarifier is designed for solid liquid separation. So the principle is the same as the first one, but when people design this secondary clarifier, they have to consider what is the mass of the bacteria, right? It's more focused on this bioreaction process. So there's also a definition called the mixed liquor. So the mixed liquor is basically the mixture in the aeration tank. Okay, so it's made of liquid, but also you have a lot of suspended solids, which are bacteria and maybe some organics that has not been removed from this primary treatment. So more specifically regarding the suspended solids, or the SS, within the amount of suspended solids, or the suspended solids in the mixed liquor is called MLSS. Okay, so it's mixed liquor suspended solids. So if you imagine, as I said, for the suspended solids, it's further composed of the bacteria and some smaller solids, which may be organics, or maybe sand, or very small chunks of solids there, which basically are not living things, right? So further, we define this bacteria section, or the microbial section, as the mixed liquor volatile suspended solids. Okay, so basically it's the MLVSS specifically targets or defines the microbial suspension in this mixed liquor. Okay, so there are quite many definitions here. We're going to use them in some calculations. Okay. So regarding the general operation of the aeration tank, as we said, the MLSS is the mixed liquor suspended solids, right? So in terms of its concentration, it's normally in the range of 2,000 to 4,000 milligram per liter. So you can see that this is a pretty high concentration, right? So when we talk about the general BOD values in the, let's say the natural water, it has to be below 8, right? So here you can see that since we're dealing with the wastewater, the concentration is very high, 2,000 to 4,000 milligram per liter. And within these suspended solids, or the MLSS, more than half of it are actually microorganisms. Okay, so what that means is that the MLVSS divided by the MLSS is higher than 50%. Okay, so you can see what is the magnitude of these bacteria inside. So actually half of them, or more than half of them, are just bacteria that we feed in to decompose these organics. So if you have a higher concentration of the MLVSS, which means that you have more bacteria, so you can just use a smaller volume of the aeration tank because it's going to be more efficient to remove these organics, right? But it's not always the higher the better. Okay, so there's always a maximum concentration, which are limited by the following things. So the first is the air requirements, because the more bacteria we have, the more air is going to consume, right? So if you don't put in enough of oxygen, then these bacteria are just not going to be able to breathe, and they're going to die, right? So the second consideration is the clarification capacity. So if you imagine, if you put in too much of the bacteria inside, MLVSS, if it's very high, okay, then what that means is that the suspended solids, or the amount of these solids, are going to be higher. And then what that means is that the secondary clarifier is going to deal with more solids in here. And potentially it's going to be an issue, because if you have too much of solids, then maybe the clarifier would not be able to deal with that. Or basically the suspended solids can get leaked out into the effluent, right? Which is another problem for the wastewater treatment. So that's why normally, or typically, the MLSS is in this range, and further the MLVSS, is, you can think of it as around half of the MLSS. Okay. So now if we zoom in to look at what is really happening in this aeration tank system, right? So here, what I'm drawing here is the aeration tank. And here is the secondary clarifier. So basically, this is going to be the effluent from the primary clarifier. Right, so as we introduced, we cannot directly pass this wastewater or the pre-treated water into the aeration tank because there are not a lot of bacteria inside, right? So we have to introduce some bacteria, which is called the activated sludge, right? To mix it with the pre-treated wastewater and then let it go to the aeration tank. So inside the aeration tank, we have the mixing and the aeration, right? Generate bubbles, right? So then after some time, or actually this is a continuous process. So within this tank, while you're stirring it, so it's like a CSTR. So the continuous stirring tank reactor, right? So you have an inlet, you have an outlet, and then basically the treated water are going to come out. So if you want to know what is the total flow of the water that's coming out of the aeration tank, then that's going to be the Q plus the QR here, because the mass of the water is going to conserve, right? So the flow rate is going to be Q plus QR. And then these treated water are going to send into a secondary clarifier, right? The secondary settling tank, so that these propagated bacteria can settle down, right? So they can basically get accumulated at the base. So basically these treated water are going to come out, sent to tertiary treatment, okay? And then at the same time, for these sludge here, so a fraction of them is sent back into the beginning to mix with the water coming out of the primary clarifier. But at the same time, a fraction of the water is just dumped into the waste. Okay, so this is the wasted sludge. And also for this fraction of sludge, we call it return sludge, because this sludge is returned to the beginning of the aeration tank, to the inlet of the aeration tank, right? So that's also the activated sludge. So now if you basically, if you set up a control volume that's restraining this area, okay, so as the dashed line box here, you can see that if we know what is the flow rate of water for the waste sludge, then we can calculate what is the effluent flow, right? So they define it as QE. Then QE is going to be equal to Q minus QW, right? Because at the inlet, we have the water coming in is equal to Q, right? So for this flow here, it's not dependent on what is the return sludge at all, right? And then at the same time, if we set up a control volume here to just look at what is the inflow and what is the outflow of this secondary clarifier here, then you can see that the inflow Q in is going to be equal to Q plus QR, right? And then for the outflow, you have the QE and you have the Q waste. And then at the same time, you have the Q return, right? So basically here you have this flow coming out to the effluent, which is the treated water. This flow coming out, this is a sludge flow, and that's split into two different flows. So one is the waste and the other is the return sludge. So you can see that here you have Q minus QW, right? And then this is going to be QW, and this is going to be QR. So these two are equal to each other, right? Because the QW is get canceled, right? So basically this is the mass balance of the water in the system. So there's also a mass balance of the suspended solids, because if you imagine, if we still choose this control volume here as the studied object, okay? So the suspended solid concentration, let's say if we know what is the suspended solid concentration here, let's define it as W, SSW. And then for the return sludge, it's going to have the same suspended solid concentration as the wastewater, as the wasted sludge, right? Because they're being split from the same flow here, okay? And then if we define the suspended solid concentration as SSE here, which is the effluent suspended solids, and further we define the concentration of the suspended solid. Let's say, actually, this is just MLSS, right? This is the concentration of the suspended solids in the mixed liquor. So then we can set up a mass balance for the suspended solids. Okay, so that's going to be in is equal to Q plus QR multiplied by MLSS, right? And then for M out, that's going to be Q minus QW multiplied by QE plus QR multiplied by SSW plus QW multiplied by SSW. Okay, so these two have to be equal to each other. Okay, so this is the mass balance for water. And this is the mass balance for the suspended solids. So we're going to use these mass balance quite a lot when we design the activated sludge system, okay? So for this aeration tank process, the dissolved oxygen concentration is going to be very important, because we have to maintain the DO at an appropriate level so that the bacteria can propagate and can also consume these organics, right? So it has to be sufficient for good BOD removal. So it's usually set as 2 milligram per liter. So again, if we recall what is the maximum BOD concentration in natural water, is 8 to 8.5 milligram per liter, right? So we are just using or maintaining at a relatively low value, because if it's too high, then it's going to cause a energy waste, because for the bacteria, if they can have a fraction of oxygen, it means that they can survive themselves, right? So because we're maintaining the concentration at a positive value. But at the same time, if the concentration is too high, then the bacteria can form these filament bacteria. What that means is, instead of forming chunks, like a cluster, they're going to form these branches, okay? These filaments, which means that it's going to be less easy or more difficult to settle down in the secondary clarifier. And at the same time, if the DO is too low, then it means that it's going to be insufficient BOD removal. And also, there may be some anaerobic decomposition. It's going to cause some odor in this process. So another design parameter is the hydraulic retention time, the HRT, or just the, people just call it aeration time. So this aeration time is similar to the sedimentation time. So basically, it's just calculated by time equal to the volume divided by Q, okay? So if you look back on this system here, so it's just the volume of the aeration tank divided by the flow rate that's going into this aeration tank. That's going to be Q plus QR, okay? So basically, it's a volume divided by Q plus QR. The basically the influent from the primary clarifier and the return sludge flow rate. So we normally control this aeration tank to 2 to 24 hours, because this propagation process of the microorganisms is relatively slow. We want to let them to grow sufficiently to remove these BOD here. So there's another definition of the time, which is called the solid retention time. So the solid retention time basically means how long these solids can stay in the entire system. So as you see earlier, these solids are being recycled, right? So while they're being settled down, while they're being propagated, they settle down in here, and then they can get returned, recycled back into the aeration tank. So we want to calculate the time that correspond to the basically the time for these bacteria to stay in the entire system. So that's why the calculation is going to be a little bit different. So the SRT, or the solid retention time, is calculated by the MLSS, the mixed liquor suspended solids, multiplied by the volume of the aeration tank. So basically, this is going to be the entire solid mass in the tank, right? So it's concentration multiplied by volume. And then in the denominator, we have the suspended solids concentration in the effluent. Remember, the E is for effluent, which is the treated water, multiplied by the flow rate of the effluent, plus the suspended solid concentration in the waste, multiplied by the flow rate in the waste. Okay, so the waste sludge. So if I draw the secondary clarifier again, we have this line is the waste, right? And this line is the effluent. So here we have Q plus QR. This is QR. So as we said, for the return sludge, it has the same concentration for the suspended solids as the waste sludge. So basically, it's just looking at what is coming out from these, really the exit of the entire aeration tank system, right? So you can see now what is the difference between the hydraulic retention time and the solid retention time. The hydraulic retention time just try to deal with the water. What is residence time of wastewater in this system? Well, the solid retention time is what is the residence time of these bacteria in the aeration tank. And because of that, this time is going to be longer compared to the water time, because the bacteria are being recycled in the system, right? So for this solid retention time, there are requirements as well, because if it's too low, it means that not enough microorganisms are being grown, right? So before they are growing into more populations, they just exit from the system. But if it's too high, then we're also going to form these filament bacteria. So basically, they form these larger branches, which is going to be difficult for them to settle down. So one important parameter is called the food to mass ratio. So the food to mass ratio, again, is using the analogy of the bacteria as enjoying endless amount of food, right? So the food is basically the influent or the inflow of the BOD, and the mass is basically the mass of the bacteria. So it's calculated, it's defined as a ratio of the influence BOD per day to the mass of the microorganisms, okay? So because of that, you can write out what is the equation, okay? So the food to mass ratio is calculated by the BOD concentration multiplied by the Q in, which is basically the, let's say the Q0, which is basically the flow rate of the wastewater coming out of the primary clarifier. Okay, so this is the influent of the BOD, and this is basically the food, right? And the mass of the microorganisms is the MLVSS multiplied by the volume, because we're specifically talking about the microorganisms, right? So those are going to be the volatile or the bacteria mass only. So now if you check what is the unit for this food to mass ratio, so the BOD has a unit of milligram per liter, right? And then the flow rate has a unit of liter per second, right? Or liter per day. So the MLVSS has a unit of milligram per liter, and then the volume has a unit of liter. So you can see that if you cancel out these terms, you see that the food to mass ratio, the F over M, has a unit of 1 per second or 1 per day. So the normal range for this food to mass ratio is 0.2 to 0.5. So this is going to be a design parameter. So basically, let's say if we know what is the BOD value coming out of the wastewater or the primary clarifier, then when we design what is the mass of the bacteria, we need to make sure that it falls in this range, right? It's going to determine how much bacteria we need to grow in this aeration tank. So further, what does this 0.2 or 0.5 mean, right? So basically, it's the food or the inflow of the food divided by the basically the weight or the mass of the bacteria. So if you can imagine a person eating, okay, so let's say let's use myself for an example. So basically, you can calculate what is the amount of food I can eat every day, right? And basically, it's the amount of food, let's say the kilogram per day. This is the inflow of the organics. And then I'll use that to divide by my weight, right? So basically, if I myself can have a food to mass ratio of 0.2, basically 20%, then what it means is that I can eat the food with around 20% of my body mass every day, which is quite significant, right? No human being can do that, at least anyone that I know of. So basically, you can see that these bacteria can really eat a lot. They can process a lot of BOD, right? So if they can go to 50%, it means that they eat half of their body mass every day, okay? So I think this is going to be a little bit easier for us to understand what this F over M ratio means. So if the value is too high, it definitely means that the bacteria cannot handle the food, because so what that means is that BOD is not going to be treated well. And also, if the food is too high, it's going to cause a process called the viscous bulking. Basically, you have too much organics, and it's going to cause less efficient settling of the bacteria. But if it's too low, then you're going to have this filament bacteria again. So you can see that the aeration tank system, since it's a biological reaction, is going to be very sensitive to a lot of parameters. So here I have two example questions. So maybe we can just quickly look at the first one. So the F over M for a treatment process, which has a flow rate of Q for this activated sludge plant is 0.2, okay? So the influent BOD after primary settling is 150. So the MLVSS is 2200 and wants us to calculate what is the volume of the aeration tank. So if it involves anything about F over M, we just write out the definition, okay? So that's going to be the BOD multiplied by Q divided by the MLVSS multiplied by volume. So if you look at this question, you have the F over M equal to 0.2, right? So the BOD is 150, flow rate is 0.4380, right? MLVSS is 2200, right? And then you're just trying to solve for V. Okay, so this is quite straightforward when we're trying to look at this aeration tank system. So there's another similar question. I think these are the two homework problems for this upcoming Wednesday. So we can further talk about these examples and then talk a little bit more about the detailed design for this aeration tank in our next class. Okay. So that's all for this class, and let me know if you have any questions. All right.